For decades, the warnings were there, quiet, technical, and easy to ignore. They were buried in academic journals, hidden inside government briefings, and spoken softly at scientific conferences that never made headlines. Long before the world learned to fear outbreaks, before lockdowns and emergency alerts became familiar, a small group of scientists were already sounding the alarm. Not about a single disease, not about one outbreak, but about something far more unsettling, a virus that was not a matter of if, but when. In the 1970s and 1980s, virologists began documenting unusual viral behaviors that did not fit existing models. These pathogens mutated faster than expected. They adapted inside the human body in ways that challenged basic assumptions of immunology. At the time, these observations were treated as anomalies, interesting, but not urgent. Funding was limited. Attention was elsewhere. The world had other priorities. Inside laboratories designed to keep danger contained, scientists worked in silence, behind thick glass, negative pressure rooms, and sealed doors marked with red biohazard symbols. Experiments were conducted with extreme caution. Researchers wore full-body protective suits not because the virus was well understood, but because it wasn't. When scientists don't fully understand a pathogen, fear becomes protocol. Internal reports from high-containment laboratories used words rarely seen in public health messaging: inevitable, uncontrollable, persistent. These were not the terms of panic, they were the terms of risk assessment. The conclusion was clear. Even then, some viruses do not behave like enemies that can be defeated. They behave more like systems evolving, adapting, and learning. By the late 1990s, classified briefings in multiple countries began outlining similar concerns. The documents did not predict a specific outbreak. Instead, they warned of a category of virus, one capable of spreading silently, mutating rapidly, and resisting long-term immunity. The threat wasn't dramatic. It was methodical, slow, relentless. One internal memo from that era contained a sentence that would later prove chillingly accurate: human response time is no longer competitive with viral evolution. At the time, this statement seemed exaggerated. Medicine was advancing rapidly. Vaccines had eradicated diseases once thought unstoppable. Antibiotics had transformed healthcare. There was confidence, perhaps overconfidence, that science would always stay one step ahead. But viruses do not follow human timelines. Unlike bacteria, viruses are not living organisms in the traditional sense. They do not eat, breathe, or reproduce on their own. Instead, they hijack the machinery of living cells, turning the body itself into a factory for replication. Every replication is an opportunity for mutation. Every mutation is a roll of the evolutionary dice. Most mutations fail. Some succeed. A few change everything. Scientists began noticing a troubling pattern. Certain viruses were no longer just mutating randomly, they were adapting strategically. Changes in their genetic structure appeared to improve transmission, evade immune responses, or prolong infection without detection. These were not viruses that burned out quickly. They lingered. They waited. One researcher described it bluntly: this is not a virus that wants to kill its host quickly. It wants to stay. That distinction matters. Historically, the most dangerous viruses were often self-limiting. They spread fast, killed fast, and eventually ran out of hosts. But a virus optimized for persistence, one that spreads before symptoms appear, one that hides from immune memory, operates under entirely different rules. By the early 2000s, warning signs were becoming harder to dismiss. Outbreaks emerged that defied containment strategies. Quarantine slowed them but did not stop them. Vaccines reduced severity but did not eliminate spread. Each response bought time but never resolution. Behind closed doors, scientists debated uncomfortable questions. What if eradication was not possible? What if containment was the best-case scenario? What if the concept of a cure itself was outdated? These questions rarely reached the public. They didn't fit into press conferences or policy slogans. Admitting uncertainty, especially at a global scale, was seen as dangerous. Reassurance was easier. Confidence was expected. But in research institutions, confidence was quietly eroding. Studies began to show that immunity, whether from prior infection or vaccination, was not permanent against this class of virus. Protection faded. Reinfections occurred. Each new cycle gave the virus more opportunities to adapt. It was not just surviving human defenses, it was learning from them. One senior virologist later reflected: we weren't racing a virus, we were training it. The most disturbing realization came when scientists understood that humanity may have already crossed a threshold. Global travel, dense urban populations, Climate-driven ecosystem shifts, all of it created ideal conditions for viral persistence. This was not the result of a single mistake. It was the outcome of decades of progress colliding with biology. The virus did not need to be engineered. It did not need malicious intent. Nature had already done the work. In high-security labs, red warning labels became routine, not because of immediate outbreaks, but because of what the data suggested over time. Trend lines told a story that individual events could not. A slow, upward curve of risk, a narrowing window for control. Some scientists tried to speak publicly. Their warnings were often softened, reframed, or misunderstood. Low probability, people heard when the real message was high impact, manageable, when the truth was unstoppable under current systems. The world preferred certainty. Science offered probabilities. And probabilities, when ignored long enough, become reality. Today, when scientists look back at those early papers, those overlooked briefings, those quiet warnings, the conclusion is unavoidable. Humanity was not unprepared because it lacked knowledge. It was unprepared because the knowledge was inconvenient. The virus they feared most was never a sudden surprise. It was a slow revelation, one that unfolded in graphs, genetic sequences, and lab notes rather than headlines. By the time the threat became visible to everyone, the conditions that allowed it to exist were already permanent. And the most unsettling part, even now, many experts believe we are still only seeing the early stages. The virus has not revealed everything it can become. And we have not yet faced the full consequences of ignoring the warning no one heard. Section 2. Not all viruses are equal. When most people hear the word virus, they imagine a single type of threat, something invisible, contagious, and eventually curable. This assumption feels logical, but it is scientifically inaccurate. In reality, viruses exist on a wide spectrum of behavior and danger. Some cause brief illness and disappear, others return seasonally, and a small number represent something far more serious: biological systems that resist long-term control. These differences are not minor details. They determine whether a virus becomes a historical footnote or a permanent part of human life. The critical mistake is assuming that deadliness equals danger. In virology, the most dangerous viruses are not always the most lethal. A virus that kills its host too quickly often limits its own spread. By contrast, a virus that keeps people alive, mobile, and unaware can move silently through populations for years. From an evolutionary perspective, survival, not destruction, is the optimal strategy. This is where fear begins. Viruses differ fundamentally from bacteria. Bacteria are living organisms. They consume energy, reproduce independently, and can be targeted without harming human cells. Antibiotics exploit these differences, attacking bacterial structures that human cells do not possess. Viruses offer no such advantage. They are not alive in the traditional sense. They exist only to enter living cells and hijack their machinery. Any attempt to destroy a virus risks damaging the host itself. This biological reality places strict limits on medicine. Antiviral drugs must walk a narrow line, aggressive enough to disrupt viral replication, yet gentle enough to avoid harming the body's own cells. This constraint alone explains why antiviral development is slow, complex, and often incomplete. But for certain viruses, the challenge goes even deeper. Some viruses mutate slowly. Their genetic material changes at a manageable pace, allowing vaccines to remain effective for decades. Others mutate rapidly, producing countless variations in a short time. Each replication cycle introduces errors. Most errors are harmless. Some weaken the virus, but occasionally, a mutation improves survival, making the virus better at spreading, hiding, or resisting immune responses. Over time, these improvements accumulate. Scientists measure this process through evolutionary pressure. Every immune response, every antiviral drug, and every vaccine becomes a selection event. Variants that survive these pressures continue, those that do not disappear. In this way, human intervention does not merely fight viruses, it shapes them. For most pathogens, this process remains slow enough to manage. For a few, it does not. Another critical factor is immune memory. Many viruses trigger strong, lasting immune responses. Once infected or vaccinated, the body remembers the invader and responds quickly to future encounters. This is the foundation of herd immunity. But some viruses interfere with this process. They alter their surface proteins just enough to avoid recognition. Others suppress immune signaling altogether. In these cases, prior exposure does not guarantee future protection. This creates a dangerous illusion of control. Reinfection is not a failure of medicine. It is a property of certain viruses. When immunity fades or becomes incomplete, the population never reaches a stable endpoint. Instead, infection becomes cyclical. Waves replace conclusions. Management replaces eradication. Over time, this shifts the entire goal of public health from elimination to endurance. Latency introduces another layer of complexity. Some viruses do not remain active after initial infection. Instead, they retreat into the body, integrating into cells or hiding in tissues beyond immune surveillance. There they wait, weeks, months, sometimes decades later, they reemerge. By the time symptoms appear, transmission may already have occurred. This strategy makes traditional containment nearly impossible. Transmission dynamics matter as much as biology. A virus that spreads only after symptoms appear can be identified and isolated. A virus that spreads before symptoms, or without symptoms at all, moves faster than detection systems. Contact tracing becomes unreliable. Testing lags behind reality. By the time action is taken, the virus has already advanced. This is not hypothetical. It is a pattern repeatedly observed. Another misconception is that technology alone can overcome these challenges. Advances in genomics, artificial intelligence, and biotechnology have transformed medicine. Scientists can now sequence viral genomes in days instead of years. They can model protein interactions at atomic resolution. They can design vaccines faster than ever before. But speed does not equal certainty. Every solution introduces new variables, new pressures, and new adaptations. For some viruses, every intervention is met with a response. This leads to a difficult scientific truth. Not all viruses are problems to be solved. Some are conditions to be lived with. The distinction is subtle but profound. Solvable problems have endpoints. Conditions persist. They require constant monitoring, repeated interventions, and continuous resources. They never truly end. From a societal perspective, this distinction changes everything. Public expectations are shaped by success stories. Smallpox was eradicated. Polio was nearly eliminated. These victories created a belief that all viruses follow the same trajectory. But these successes were possible because those viruses shared specific vulnerabilities. They mutated slowly. They produced visible symptoms. They lacked animal reservoirs. They did not hide inside hosts for long periods. The viruses scientists fear most share none of these weaknesses. They exist across species. They move between animals and humans. They adapt to immune pressure. They persist within populations. They exploit global connectivity. And most importantly, they do not require catastrophic outcomes to remain successful. Their danger lies in continuity, not collapse. Over time, this reshapes the role of medicine. Doctors no longer aim to cure every case. Instead, they reduce severity. They prevent hospital overload. They buy time. These are meaningful achievements, but they are not victories in the traditional sense. This reality is rarely communicated clearly. Public messaging favors reassurance. Complexity is reduced to slogans. Uncertainty is softened. But within the scientific community, the conversation is different. Researchers understand that certain viruses represent a permanent negotiation between biology and civilization, and that negotiation is not always favorable to humans. As populations grow denser and ecosystems change, opportunities for viral evolution increase. Climate shifts expand the range of vectors. Global travel collapses distance. Urbanization concentrates hosts. Each factor amplifies the others. What once took centuries can now happen in years. This is why scientists do not fear viruses equally. They assess threat based on adaptability, persistence, and unpredictability. A virus that cannot be cured but can be controlled is manageable. A virus that resists both cure and control is something else entirely. That distinction is at the heart of the fear, because once a virus enters that category, the question is no longer how to stop it. The question becomes how long humanity can stay ahead of it, and whether staying ahead is even possible. Section 3. The pattern behind every global outbreak. History rarely repeats itself exactly, but in the case of global outbreaks, it follows a recognizable pattern. Each time a major virus emerges, the details appear different: new locations, new symptoms, new names, but beneath the surface, the structure remains the same. Scientists have spent decades mapping these patterns, not to predict the next outbreak precisely, but to understand why outbreaks keep happening despite everything humanity learns in between. The first stage is always invisibility. Long before a virus becomes a headline, it exists quietly at the margins of human awareness. It circulates in animal populations, mutates in isolated hosts, or spreads unnoticed among small human communities. During this phase, there are no alarms because there is nothing obvious to detect. The virus is present, but it has not yet crossed the threshold where society recognizes it as a threat. This stage can last years, sometimes decades. When spillover occurs when a virus moves from animals into humans, it is rarely dramatic. The initial infections are often mild, misdiagnosed, or ignored. Symptoms resemble common illnesses. Cases appear isolated. No clear pattern emerges. From a statistical perspective, the signal is buried in noise. Surveillance systems are not designed to react to ambiguity, and so the virus is allowed time. Time is the most valuable resource a virus can have. As transmission increases, the second stage begins: misinterpretation. Doctors see clusters of illness but attribute them to known causes. Health agencies issue cautious statements. Early data is incomplete, sometimes contradictory. This uncertainty delays decisive action. Every delay allows the virus to spread further, diversify genetically, and establish itself in new regions. This stage is where containment often fails. The third stage is recognition without readiness. At this point, the virus can no longer be ignored. Case numbers rise. Hospitals notice patterns. Researchers isolate the pathogen and begin studying its structure. Governments acknowledge the threat publicly, but recognition does not equal control. Scientific understanding lags behind viral spread. By the time models are built, the virus has already moved. This gap between awareness and action is a constant across outbreaks. The fourth stage is intervention. Travel restrictions are imposed. Public health measures are introduced. Laboratories race to develop diagnostics, treatments, and vaccines. These efforts slow the virus, but rarely stop it outright. Interventions reduce impact, not existence. The virus adapts to new pressures, selecting variants better suited to survive in a world that is now actively resisting it. At this point, the virus and humanity enter a feedback loop. Each human response reshapes the viral landscape. Each viral adaptation reshapes human strategy. This cycle repeats continuously. The idea of a single decisive victory becomes unrealistic. Instead, progress is measured in reduced severity, fewer deaths, and delayed peaks. These outcomes matter deeply, but they also mark a shift from eradication to management. The final stage is normalization. Over time, societies adapt. Emergency measures fade. The virus becomes part of the background. Surveillance continues, but attention moves on. New generations grow up knowing the virus as a fact of life rather than a crisis. This transition does not mean the virus is gone. It means humanity has adjusted its expectations. Every major outbreak in modern history follows this arc. What changes is not the pattern, but the speed. Global connectivity has compressed timelines. What once unfolded over decades can now happen in months. International travel allows a localized spillover event to become a global phenomenon before detection systems activate. Dense urban environments provide ideal conditions for sustained transmission. Supply chains, migration, and climate-driven ecological changes further accelerate spread. These factors do not create viruses. They create opportunities. Scientists studying outbreak patterns have identified a troubling trend. Each successive global event becomes harder to control than the last. Not because medicine is failing, but because the environment in which viruses operate has fundamentally changed. Humanity has become more interconnected, more mobile, and more densely packed than at any point in history. From a viral perspective, this is optimal. Another recurring pattern is over-reliance on past success. Strategies that worked against previous viruses are often applied too confidently to new ones. This assumption delays innovation. It also underestimates the uniqueness of each pathogen. While viruses share broad characteristics, small differences in transmission timing, mutation rate, or immune interaction can completely alter outcomes. One size does not fit all in virology. There is also a psychological pattern that repeats with every outbreak. Early warnings are dismissed as alarmist. Mid-stage responses are criticized as insufficient or excessive. Late-stage acceptance reframes the event as unavoidable. This cycle influences policy decisions, public trust, and scientific communication. It shapes not only how outbreaks unfold, but how they are remembered. And memory matters. When lessons are not institutionalized, they fade. Funding decreases. Preparedness plans gather dust. Expertise disperses. The next outbreak then appears unprecedented, even when it follows a familiar structure. This creates the illusion of surprise when in reality, the conditions were already in place. For the virus scientists fear most, this pattern is especially dangerous because it does not rely on dramatic emergence. It does not require visible devastation to succeed. It benefits from delay, uncertainty, and normalization. Each stage strengthens its position. Each cycle embeds it deeper into human systems. The most unsettling insight from outbreak science is this: Humanity is not facing isolated viral threats. It is facing an era shaped by repeated viral pressure. The question is no longer whether another outbreak will occur. The question is how many will overlap, and how resilient global systems truly are. In this context, the virus scientists fear the most is not an anomaly. It is an expression of the pattern itself, optimized by evolution, amplified by modern life, and sustained by the very structures that define progress. Understanding this pattern does not eliminate the threat, but failing to understand it guarantees repetition. And repetition, in biology, always favors the organism that adapts fastest. Section 4. The virus that doesn't play by the rules. Most viruses follow predictable biological constraints. They infect, replicate, spread, and eventually encounter limits imposed by immunity, treatment, or population dynamics. These constraints form the foundation of modern epidemiology, but the virus scientists fear most does not remain within those boundaries. It behaves in ways that challenge assumptions built over decades of research. It does not simply exploit weaknesses in human systems, it exposes weaknesses in biological understanding itself. One of the first rules it breaks is the relationship between infection and detection. Traditional disease control relies on visibility. Symptoms signal infection. Testing confirms it. Isolation interrupts spread. This virus disrupts that sequence. Infection often occurs long before symptoms, and in many cases, symptoms never appear at all. Transmission happens silently, efficiently, and continuously, rendering symptom-based surveillance fundamentally incomplete. From an evolutionary standpoint, this is an extraordinary advantage. Another rule involves mutation. While all viruses mutate, most do so within a constrained range that preserves their basic structure. Excessive mutation usually weakens a virus, damaging the proteins it needs to survive. But this virus operates near the edge of instability without collapsing. Its replication machinery tolerates errors at a rate that would destroy most pathogens. Instead of degrading, it diversifies. This creates not one virus, but a constantly shifting population. Within a single host, thousands of slightly different versions can exist simultaneously. This internal diversity allows rapid adaptation to immune pressure. When antibodies neutralize one variant, others persist. When treatments suppress replication, resistant forms emerge. The virus does not wait for population-level evolution. It evolves inside individuals. This fundamentally alters the timeline of adaptation. Another broken rule concerns immune memory. The human immune system relies on recognition. Once it identifies a pathogen, it stores information for faster response in the future. This process underlies vaccination and natural immunity. The virus scientists fear most interferes with this mechanism in subtle but profound ways. It alters key surface proteins, just enough to evade recognition without sacrificing functionality. The immune system responds but not decisively. Over time, this leads to partial immunity rather than sterilizing immunity. Infections become less severe, but not impossible. Transmission continues, even among previously exposed populations. This creates a stable reservoir of infection that never fully disappears. The virus becomes endemic, embedded within society rather than eliminated from it. Another assumption that fails involves host specificity. Many viruses are limited to a narrow range of species. This limits their evolutionary options and simplifies control. The virus in question does not respect this boundary. It moves between species, adapting to different cellular environments with minimal loss of efficiency. Animal reservoirs allow it to persist outside human populations, immune to human interventions. This makes eradication biologically unrealistic. Even when human transmission declines, the virus remains active elsewhere, evolving beyond reach. When conditions allow, it re-enters human populations with new characteristics. Each return introduces uncertainty. Prior data becomes less reliable. Models lose accuracy. Planning becomes reactive rather than preventive. The virus also disrupts assumptions about severity and spread. Traditionally, higher transmissibility is associated with lower severity, and vice versa. This trade-off limits viral impact. The virus scientists fear most does not adhere strictly to this balance. It maintains efficient spread while retaining the capacity for severe outcomes under specific conditions. This unpredictability complicates risk assessment. Severity varies widely between individuals. Age, genetics, immune history, and environmental factors all influence outcomes. This variability masks danger. Early data may underestimate risk. Later waves reveal consequences that were not apparent initially. By the time patterns stabilize, the virus has already established itself. Another broken rule involves treatment resistance. Antiviral drugs target specific stages of replication. For many viruses, this creates a bottleneck that limits escape. In this case, the virus uses multiple overlapping pathways. Blocking one route slows it, but rarely stops it entirely. Combination therapies help, but resistance emerges over time. This forces continuous drug development. Unlike antibiotics, which can remain effective for decades, antivirals face rapid obsolescence against highly adaptive viruses. This creates a perpetual race. Scientific progress becomes maintenance rather than resolution. The goal shifts from victory to sustainability. The virus also exploits social systems as effectively as biological ones. It spreads through routine human behavior: work, travel, caregiving, social interaction. Controlling it requires altering daily life at scale. Such measures carry economic, psychological, and political costs. Over time, compliance erodes. Fatigue sets in. The virus benefits from this predictably human response. Biology and behavior intersect. Perhaps the most unsettling rule it breaks is the assumption of containment. Traditional outbreak models assume that with sufficient coordination, spread can be localized. Borders, quarantines, and targeted interventions can limit impact. This virus consistently escapes such boundaries. It moves faster than decision-making processes. By the time containment is attempted, it has already succeeded. This creates a sense of inevitability. For scientists, this combination of traits is deeply troubling. Each characteristic alone would be manageable. Together, they form a system optimized for persistence in a modern world. The virus does not need to overwhelm healthcare systems continuously. It only needs to remain present, adapting quietly, waiting for opportunities. This is why experts struggle to communicate the threat. It does not conform to cinematic expectations of disaster. There is no single moment of collapse. Instead, there is gradual normalization of risk, a slow recalibration of what is considered acceptable loss. From a scientific perspective, this represents a shift in the relationship between humanity and infectious disease. Control is no longer absolute. Prediction is no longer reliable. The margin for error narrows with each cycle of adaptation. And this leads to the central realization. The virus scientists fear most is not dangerous because it is unstoppable in theory. It is dangerous because stopping it would require perfect coordination, permanent vigilance, and biological control that exceeds current limits. Nature does not require perfection. Humans do, and that imbalance defines the threat. Section 5. Why a cure may be scientifically impossible. The word cure carries an assumption. It suggests an endpoint, a moment when a disease is removed, a threat eliminated, and normality restored. In modern medicine, this expectation is deeply ingrained. Cancers are cured, infections are cured, symptoms resolve, and life moves forward. But this framework does not apply universally. In virology, the concept of a cure is not a default outcome. It is an exception, and for some viruses, it may not exist at all. To understand why, it is necessary to confront an uncomfortable truth. Medicine does not defeat viruses in the way people imagine. The immune system does most of the work. Drugs and vaccines do not directly eliminate viruses. They assist the body in controlling them. When the immune system fails to achieve complete clearance, medicine has no independent mechanism to finish the job. This limitation is structural, not technological. A virus does not live on the surface of the body. It lives inside cells. Once inside, it becomes part of the cellular environment, using the host's own machinery to replicate. Any intervention that fully destroys the virus must either disrupt this machinery or remove the infected cells themselves. Both options carry risk. Cells are not disposable. They form tissues, organs, and systems. Eliminating infected cells at scale can cause irreversible damage. This is why antiviral treatments are inherently conservative. They slow replication. They reduce viral load. They alleviate symptoms, but they rarely achieve total elimination. The closer a treatment comes to complete suppression, the narrower the margin of safety becomes. At a certain point, toxicity replaces efficacy. This trade-off defines the boundary of possibility. Another barrier lies in viral integration. Some viruses do not merely occupy cells temporarily. They integrate genetic material into the host's DNA or persist in cellular reservoirs that are invisible to immune surveillance. Once this occurs, the virus is no longer an external invader. It is embedded within the body itself. Removing it would require editing or destroying the host's own genome or tissues. From a medical standpoint, this is indistinguishable from self-harm. Even when integration is incomplete, persistence creates similar challenges. Certain tissues such as the nervous system, reproductive organs, and immune-privileged sites are shielded from aggressive immune responses. These regions evolve to protect vital functions from inflammation. Viruses that establish presence in these areas gain long-term protection. Drugs struggle to reach them. Immune cells are restricted from acting fully. These reservoirs function as biological safe houses. A cure would require reaching every infected cell simultaneously. Missing even a small fraction allows resurgence. Viral replication does not need perfection. Human intervention does. This asymmetry heavily favors the virus. Mutation further complicates the problem. A cure assumes a stable target, but for highly adaptive viruses, the target is constantly moving. Proteins change shape. Entry mechanisms evolve. Replication pathways diversify. A drug designed for one configuration becomes less effective against another. Combination therapies attempt to address this by targeting multiple stages at once, but each additional target increases complexity and risk. The more comprehensive the intervention, the more fragile it becomes. Vaccines face parallel constraints. They do not cure existing infections. They prepare the immune system for future encounters. For viruses with stable antigens, this approach is highly effective. For viruses that alter those antigens frequently, immunity becomes partial and temporary. Booster strategies extend protection but do not resolve the underlying instability. This creates a cycle of continual adaptation. Some have proposed advanced technologies as solutions. Gene editing, personalized immunotherapy, synthetic antibodies. These approaches show promise in controlled settings, but scalability remains a barrier. A cure must work not for individuals, but for populations. It must be safe across genetic diversity, age groups, and health conditions. It must be deployable at global scale. It must be affordable. It must be repeatable. Every requirement narrows feasibility. There is also the issue of timing. A cure administered after widespread infection must overcome entrenched viral diversity. Early intervention could, in theory, limit this. But early intervention requires early detection. For a virus that spreads silently and rapidly, detection always lags behind reality. By the time a cure exists, the virus has already diversified beyond a single solution. This is not pessimism, it is mathematics. The immune system itself introduces further limitations. Chronic viral presence can exhaust immune responses. T cells lose effectiveness. Inflammatory pathways become dysregulated. In some cases, the immune system contributes to pathology rather than resolution. Treatments must then balance viral suppression with immune modulation. The goal shifts from elimination to equilibrium. At this stage, cure becomes a misleading term. The final constraint is evolutionary. Viruses existed long before humans. They will exist long after. Eradication is rare because it requires a combination of biological vulnerability, human coordination, and environmental stability that almost never align. When eradication fails, persistence is not a failure of science. It is the default outcome of natural selection. From this perspective, the question changes. Instead of asking why there is no cure, scientists ask why one should exist at all. Nature does not design systems for human convenience. It selects for survival. A virus that cannot be cured is not broken. It is successful. This realization reframes the fear. The virus scientists fear most is not dangerous because science has failed. It is dangerous because science is confronting its limits. The tools available can mitigate harm, extend life, and reduce suffering, but they cannot rewrite fundamental biological rules. Humanity has grown accustomed to solutions. In this case, the solution may not be elimination, but adaptation, continuous surveillance, repeated intervention, behavioral change, medical management without final resolution. This is not a defeat, but it is not a victory either. It is a permanent condition. And once a virus enters this category, the world does not return to what it was before. It moves forward under new constraints. Section 6. The laboratories racing against time. Behind every public health announcement, every policy decision, and every headline, there exists a quieter world where the real struggle takes place. It is not a world of press conferences or political debates. It is a world of sealed laboratories, controlled airflows, redundant safety systems, and scientists working against biological clocks that never stop. This is where humanity confronts the virus directly, not as a social problem, but as a physical reality. The highest risk research occurs in laboratories designed to contain what cannot be allowed to escape. These facilities operate under extreme conditions. Air pressure flows inward. Waste is sterilized multiple times. Entry and exit procedures are rigid, repetitive, and unforgiving. Every protocol exists because something once went wrong somewhere else. In these environments, caution is not optional. It is the price of proximity. Scientists working here understand something the public rarely sees. Knowledge does not arrive fully formed. It is assembled slowly, through fragments of data, failed experiments, and incremental insights. Each step forward is measured against safety. Speed is demanded by the threat, but restraint is demanded by reality. This tension defines every decision. Modern virology relies heavily on genetic sequencing. When a new viral sample arrives, its genome can be mapped in hours. Variants can be tracked in near real time. This capability has transformed surveillance, allowing scientists to observe evolution as it happens. But observation is not control. Seeing change does not prevent it. Often, it merely confirms how fast adaptation is occurring. Drug discovery operates on a longer timeline. Identifying a molecular target is only the beginning. Compounds must bind precisely. They must disrupt viral processes without disrupting cellular ones. Promising candidates frequently fail not because they are ineffective, but because they are unsafe. Toxicity eliminates more treatments than inefficacy ever does. This is not a flaw in research. It is the cost of biology. Even when treatments succeed in early trials, scale introduces new problems. What works in controlled environments may behave differently across populations. Genetic diversity alters responses. Pre-existing conditions complicate outcomes. Interactions with other medications introduce risk. Each variable expands uncertainty. Each uncertainty slows deployment. Vaccines follow a parallel path. Designing one is not the same as proving durability. Short-term protection can be demonstrated relatively quickly. Long-term immunity cannot. It requires time, sometimes years, to understand how protection wanes, how variants escape, and how immune memory evolves. During that time, the virus does not pause. It continues adapting under selective pressure. Artificial intelligence has accelerated parts of this process. Models predict protein folding. Algorithms screen vast libraries of compounds. Simulations narrow the search space. These tools increase efficiency, but they do not eliminate fundamental constraints. Predictions must still be tested in living systems. Biology remains the final authority. There is also an ethical boundary laboratories cannot cross. Human challenge studies, deliberate infection, are limited or prohibited for viruses with uncertain outcomes. This restricts experimental design. Scientists cannot simply test hypotheses at will. They must wait for natural data to emerge, even when time is critical. Safety outweighs speed. Funding introduces another pressure. Research priorities shift with public attention. Emergency funding surges during crises and recedes afterward. Long-term projects struggle for continuity. Teams disperse. Infrastructure ages. When the next wave arrives, capacity must be rebuilt. This cycle weakens preparedness and amplifies urgency. International collaboration helps, but it is imperfect. Data sharing depends on trust, transparency, and political alignment. Delays in reporting slow global response. Differences in regulation complicate joint trials. Scientific cooperation exists within geopolitical reality, not above it. Within laboratories, scientists confront another challenge: uncertainty itself. Early data is incomplete. Signals conflict. Results change as sample sizes grow. Decisions must be made without definitive answers. This environment favors caution, but caution can look like hesitation from the outside. The gap between scientific process and public expectation widens. Meanwhile, the virus continues to replicate. Every replication is an experiment conducted by nature. Billions occur daily. Most fail, some succeed. The virus explores genetic space at a scale no laboratory can match. Human research is deliberate and constrained. Viral evolution is relentless and opportunistic. This imbalance shapes outcomes more than any single breakthrough. Scientists understand this asymmetry. They do not expect to outpace evolution permanently. Instead, they aim to narrow the gap, to slow adaptation, to anticipate changes, to reduce impact. Success is measured in margins, not absolutes. A few percentage points of effectiveness matter. A few weeks of delay matter. But these gains come at a cost. Working under constant urgency takes a psychological toll. Burnout is common. Errors become more likely as pressure increases. Safety incidents, even minor ones, reinforce fear. Researchers carry responsibility not only for discovery, but for containment. Failure has consequences beyond careers. Despite this, the work continues. Every new dataset refines understanding. Every failed trial informs the next. Knowledge accumulates even when solutions do not. This is how science advances, not through sudden victories, but through sustained resistance against uncertainty. The laboratories racing against time are not searching for a miracle. They are searching for leverage, small advantages that shift balance, better diagnostics, faster response, broader protection. These tools do not eliminate the virus, but they change how society lives with it. And that distinction matters, because the race is not between humanity and a single pathogen. It is between human systems and biological processes that never stop optimizing. The laboratories represent the front line of this race, but they do not control its direction alone. They can slow the clock. They cannot stop it. Section 7. The moment scientists admitted fear extended in depth. For most of modern scientific history, uncertainty was treated as a temporary condition. Data would improve, models would sharpen, variables would narrow. The assumption was not arrogance, but experience. Again and again, science had turned unknowns into knowns, chaos into structure. This expectation shaped not only research methods, but identity. Scientists were problem solvers by definition. What unsettled them in this case was not ignorance but completion. By the time fear began to surface, the data was no longer missing. Sequencing was comprehensive. Transmission pathways were mapped. Immune responses were characterized in detail. The virus was no longer mysterious in the traditional sense. And yet, the conclusions drawn from this clarity were worse than ignorance. The more complete the picture became, the less room there was for optimism. This is when fear stopped being emotional and became analytical. One of the earliest cracks appeared in internal review panels. These were not emergency meetings, but routine evaluations, places where optimism is normally rewarded with skepticism. Yet increasingly, proposals aimed at eradication were quietly downgraded. Language shifted from eliminate to reduce, from prevent to delay. Reviewers were not rejecting ambition. They were responding to evidence. The fear was embedded in the footnotes. Another fracture emerged in epidemiological forecasting. Early in outbreaks, models are expected to be wrong. They are crude tools, built quickly, but as time progressed, even refined models struggled, not because of computational limits, but because human behavior and viral evolution interacted in ways that resisted stable prediction. Interventions changed outcomes, which changed behavior, which changed viral dynamics, which invalidated prior assumptions. The system never settled. Scientists are trained to trust convergence. When multiple independent lines of evidence point to the same conclusion, confidence increases. In this case, convergence pointed not toward a solution, but toward permanence. Genomic data, clinical outcomes, reinfection rates, immune waning, and ecological reservoirs all told the same story. This virus was not burning out. That realization carried weight. For clinicians, fear manifested differently. Hospitals adapted. Protocols improved. Mortality declined. On the surface, this looked like success, but beneath it lay exhaustion. Waves did not end. They spaced out. Recovery time shortened. The crisis became chronic. Doctors began speaking privately about sustainability rather than survival. Sustainability is not a word used when victory is expected. Immunologists faced a particularly disturbing insight. The immune system was not failing catastrophically. It was responding exactly as designed, controlling severe disease while allowing persistent circulation. From an evolutionary standpoint, this was logical. From a public health standpoint, it was devastating. The immune system was cooperating with the virus just enough to keep both alive. This was not a flaw, it was balance. Fear deepened when long-term consequences became visible. Not mass death, but cumulative damage, repeated infections, chronic inflammation, subclinical effects that did not trigger alarms but slowly eroded health. Scientists realized that even mild outcomes were not neutral when multiplied across populations and time. The cost was diffuse, delayed, and therefore easy to ignore. Institutional fear emerged when contingency planning failed to produce endpoints. Emergency frameworks are built around resolution. They assume escalation followed by de-escalation. When de-escalation never fully arrives, institutions strain. Funding cycles break alignment. Political will fragments. Scientific recommendations become harder to enforce. This exposed a weakness unrelated to biology. Another moment of admission came when experts compared this virus to past successes. Smallpox, polio, measles. The contrast was stark. Those viruses lacked asymptomatic spread, rapid mutation, and animal reservoirs. They were vulnerable in ways this virus was not. The comparison itself became evidence that the current threat belonged to a different class. Fear arose from classification. Behind closed doors, some scientists began to articulate what could not be said publicly, that humanity might be entering a long-term coexistence with a pathogen that continuously reshapes risk. Not a single enemy, but a shifting pressure always present, always adapting, never decisive. This was not apocalypse, it was erosion. The most profound fear was not about mortality rates or hospital capacity. It was about precedent. If one virus could establish this pattern, others could follow. The infrastructure enabling the success, global mobility, ecological disruption, dense populations was not temporary. It was structural. This virus was not an exception. It was a preview. That realization landed heavily. Fear also emerged from the limits of persuasion. Scientists could produce evidence, but not compliance. They could recommend strategies, but not enforce them. Over time, public trust fluctuated. Polarization distorted messaging. Expertise itself became contested terrain. The virus exploited this indirectly, benefiting from confusion and fatigue. This was beyond laboratory control. When scientists admitted fear, it was not in statements or speeches. It was in pauses, in the removal of timelines, in the cautious avoidance of promises, in the shift from declarative language to conditional phrasing. The absence of certainty became the message. Fear, in this context, was the recognition that the problem was no longer purely scientific. It was systemic. The virus had revealed vulnerabilities not just in immune systems, but in coordination, communication, and endurance. Science could slow it, medicine could blunt it, but no discipline alone could resolve it. This was the breaking point, not because experts lost faith in science, but because they finally understood what science was telling them. The virus scientists feared most was not the one they did not understand. It was the one they understood completely and still could not end. Section 8. The virus scientists fear the most reveal. The virus scientists fear the most is not defined by lethality. It is not the fastest spreading, the most visible, or even the most politically disruptive. Those traits attract attention, funding, and urgency. The virus scientists fear the most is quieter. It does not announce itself with immediate catastrophe. It settles in, it adapts, it becomes familiar before it becomes dangerous. And by the time it is understood, it is no longer something that can be removed without dismantling the systems it now inhabits. This fear emerges from a realization that took years to crystallize. The most successful virus is not one that overwhelms humanity, but one that integrates into it. Evolution does not reward destruction, it rewards persistence. The virus scientists fear the most is one that learns how to remain just below the threshold of collective alarm, causing enough harm to spread efficiently, but not enough to provoke unified global action. This balance is not accidental. It is the outcome of selective pressure operating at planetary scale. What makes this virus different is not a single mutation or mechanism, but a convergence of properties that reinforce one another. It spreads efficiently before symptoms appear. It mutates incrementally rather than explosively. It produces immunity that is real but incomplete. It reinfects without dramatic escalation. It exploits human behavior not through manipulation, but through normality, routine movement, routine contact, routine fatigue. Each feature alone is manageable. Together, they form a system that resists resolution. Scientists once believed that widespread immunity would represent an endpoint. Either through infection, vaccination, or both, the population would eventually become inhospitable to sustain transmission. But this virus altered that assumption. Immunity reduced severity, not circulation. Protection faded gradually. Reinfection became expected rather than exceptional. The virus did not need to defeat the immune system. It only needed to negotiate with it. This negotiation is what alarms immunologists the most. The immune response is not being overwhelmed or bypassed. It is being engaged and tolerated. The virus provokes just enough response to ensure survival of the host, while maintaining enough replication to ensure its own continuity. From a biological perspective, this is near optimal adaptation. From a public health perspective, it is a nightmare scenario. The fear deepens when this behavior is projected forward. Each cycle of infection provides an opportunity for refinement, not radical transformation, but tuning. Slight changes in receptor binding, minor shifts in immune evasion, subtle alterations in tissue preference. Over time, these adjustments compound. The virus does not need to become deadlier to become more consequential. It only needs to become harder to notice. One of the most unsettling discoveries came from longitudinal studies. Researchers began tracking outcomes not in weeks or months, but in years. They observed that even when individual infections appeared mild, the cumulative impact was not neutral. Repeated immune activation, low-grade inflammation, vascular stress, neurological effects, none of these triggered immediate alarms, but together, they suggested a slow erosion of baseline health across populations. This raised a question science was not prepared to answer cleanly. What does damage look like when it is distributed, delayed, and statistically invisible at the individual level, but significant in aggregate. Traditional metrics failed. Hospitalizations declined, mortality stabilized, yet disability claims rose. Workforce participation shifted, cognitive and cardiovascular baselines subtly changed. The virus was no longer a crisis. It was a background condition. The virus scientists fear the most is one that transforms public health from an episodic challenge into a permanent management problem. Not something that can be eliminated, but something that must be continually mitigated. This changes the relationship between society and disease. Resources are no longer mobilized temporarily, they are drained indefinitely. Attention wanes, compliance weakens, fatigue becomes structural. Animal reservoirs intensified this fear. When researchers confirmed sustained transmission across multiple species, the implications were immediate and irreversible. Even perfect human control would not be sufficient. The virus could retreat, evolve, and re-emerge without warning. This removed the concept of a clean victory entirely. Eradication ceased to be a scientific goal and became a theoretical impossibility. This is the moment the reveal becomes unavoidable. The virus scientists fear the most is not a future pathogen, is the one already embedded in global systems: ecological, economic, biological, and social. It has learned how humans move, how they gather, how they resist inconvenience, how they politicize uncertainty. It does not need intention. The environment does the work for it. There is fear in how predictably this virus exploits division, not because it creates misinformation, but because it benefits from it. Confusion slows response. Debate delays action. Inconsistency creates openings. The virus thrives not in ignorance, but in disagreement. Scientists recognized that even perfect data could not overcome fractured trust. The problem was no longer just biological, it was epistemological. The most disturbing realization came when experts stopped asking how to defeat the virus and began asking what kind of future it selects for. A world in which illness is normalized, where productivity adjusts downward quietly, where chronic conditions expand invisibly, where health disparities widen not through dramatic collapse, but through persistent pressure. The virus does not need to collapse systems to reshape them. This is why fear did not arrive as panic. It arrived as resignation tempered by responsibility. Scientists did not stop working. They refined treatments. They updated vaccines. They improved surveillance. But the tone changed. The language shifted away from endpoints. Words like control, management, and adaptation replaced elimination. This was not surrender. It was acknowledgement. The virus scientists fear the most forces humanity into a long-term negotiation with biology, one that has no final agreement. Every gain must be defended repeatedly. Every success decays over time. Progress becomes temporary by default. This is exhausting not only logistically, but psychologically. Humans are not built for indefinite vigilance. And then there is the precedent. If one virus can achieve this balance, others can too. Climate change, habitat disruption, and global connectivity are accelerating opportunities for similar adaptations. This virus is not the anomaly. It is the prototype. Scientists understand this clearly, even when it is not spoken aloud. The reveal, ultimately, is not about a name or a strain. It is about a category of threat that does not conform to historical narratives of victory or defeat. The virus scientists fear the most is one that aligns too well with the modern world: fast enough to spread, slow enough to endure, mild enough to be tolerated, complex enough to resist resolution. It is feared not because it is unstoppable, but because stopping it would require changes that extend beyond medicine. Changes in behavior, in governance, in global cooperation, in how societies value prevention over reaction. These are not scientific challenges alone, they are civilizational ones. By the time this realization fully set in, the fear was no longer urgent. It was settled, a quiet understanding shared among experts that the question was no longer whether humanity could defeat this virus, but whether it could adapt without losing resilience, equity, and trust in the process. The virus scientists fear the most has already taught them something uncomfortable, that the greatest biological threats of the future may not arrive as sudden disasters. They may arrive as systems that never quite leave. Section 9. What happens if IT evolves again? The most dangerous assumption humanity can make at this stage is that evolution is something that happens in bursts, separated by long periods of stability. In reality, evolution never pauses. It does not wait for permission, consensus, or readiness. It proceeds incrementally, continuously, and invisibly, shaped by pressures that humans often fail to recognize as selection forces. When scientists ask what happens if this virus evolves again, they are not asking an abstract question. They are acknowledging a certainty. The only uncertainty lies in direction, speed, and consequence. Evolution does not require intent. It does not require dramatic mutation. Most evolutionary change occurs through small adjustments that, individually, appear inconsequential. A slightly higher binding affinity, a marginal improvement in replication efficiency, a subtle delay in immune recognition. These changes are rarely headline-worthy, yet when they accumulate across billions of infections, they reshape the biological relationship between host and virus. The future, in this context, is not a surprise event. It is a statistical outcome. The probability landscape is shaped by exposure. Every infection is an experiment. Every replication cycle is a chance. The more widely and continuously a virus circulates, the more opportunities it has to explore genetic space. This is not speculation. It is a fundamental principle of evolutionary biology. The virus does not need to want to become better adapted. The environment selects for variants that persist, and persistence itself becomes the dominant advantage. One of the most critical misunderstandings in public discourse is the belief that evolution necessarily trends toward greater severity. In reality, severity is often a liability. A virus that incapacitates its host too quickly limits its own transmission. What evolution tends to favor instead is efficiency. Efficiency in entry, efficiency in spread, efficiency in coexistence. The most probable future is not one defined by sudden collapse, but by deeper entrenchment. Scientists modeling future evolutionary paths do not focus on extreme scenarios, because extremes are statistically rare. They focus on convergence, on the likelihood that the virus will continue refining its ability to reinfect previously exposed populations, on the probability that immune escape will remain partial rather than absolute, on the expectation that symptom profiles may shift rather than intensify. These are not dramatic changes, but they are destabilizing ones, because they resist decisive intervention. One highly probable evolutionary direction is increased specialization in immune evasion. This does not mean complete escape. Complete escape would provoke strong selective pressure for new countermeasures. Partial evasion is more advantageous. It allows the virus to circulate within populations that believe themselves protected, reducing behavioral caution, while maintaining transmission. From an evolutionary standpoint, this is a stable strategy. Another likely trajectory involves changes in tissue preference. Small shifts in which cells are most efficiently infected can alter disease patterns without increasing apparent severity. A virus that favors upper respiratory replication spreads more easily. One that intermittently affects vascular or neurological systems may produce delayed effects that are difficult to attribute directly. These shifts do not announce themselves. They are discovered retrospectively, through patterns rather than events. Probability also favors gradual shortening of intervals between reinfections. As immune memory wanes and viral variants adjust, the cycle compresses. This does not overwhelm healthcare systems immediately. Instead, it increases baseline illness frequency. Productivity loss becomes normalized. Chronic inflammation becomes statistically visible only at scale. Individually, people recover. Collectively, capacity erodes. Scientists are particularly concerned about evolutionary paths that intersect with aging populations. Immune systems do not remain static over a lifetime. As global demographics skew older, selection pressures change. A virus that causes tolerable outcomes in younger hosts, but compounds risk in older ones does not need to change to increase its impact. The environment does that work. Probability, not mutation, drives the outcome. Another future scenario with high likelihood involves recombination. When multiple variants circulate simultaneously, genetic material can be exchanged during co-infection. This is not hypothetical. It is a documented mechanism. Recombination does not require dramatic leaps. It allows the virus to combine advantageous traits already tested by selection. This accelerates adaptation without introducing instability. The risk here is not the emergence of a radically different virus, but the consolidation of features that increase persistence. Better immune tolerance combined with higher transmissibility. Broader host range combined with reduced symptom severity. Each combination moves the virus further away from eradication and closer to permanence. The probability framework also forces scientists to confront uncomfortable trade-offs. Interventions themselves become selective pressures. Widespread immunity shifts evolutionary advantage toward variants that can circulate despite it. Antiviral treatments select for resistance. Even behavioral changes alter the landscape. None of these are arguments against intervention. They are reminders that intervention reshapes evolution rather than stopping it. What alarms researchers is not the possibility of a single disastrous mutation, but the inevitability of incremental drift combined with social fatigue. Over time, vigilance declines, surveillance weakens, data gaps widen. The virus does not need to outpace science. It only needs to wait for attention to move elsewhere. The future shaped by probability is one in which outbreaks become harder to define. Waves blur into background noise, metrics lose clarity. Policymakers struggle to justify responses to a threat that does not peak dramatically. This is not because the threat has vanished, but because it has distributed itself across time and populations. Long-term modeling suggests that the most likely consequence of continued evolution is not collapse, but constraint. Healthcare systems operate closer to capacity more often. Economic growth absorbs hidden losses. Educational outcomes fluctuate subtly. These are not catastrophic outcomes in isolation. Together, they redefine what normal looks like. Scientists also consider the probabilistic interaction with other pathogens. Co-infections alter immune responses. Sequential infections reshape vulnerability. A virus that evolves in isolation behaves differently from one evolving within a crowded microbial ecosystem. As global travel and climate change increase pathogen overlap, these interactions become more frequent. The future is not linear. It is combinatorial. There is also the question of evolutionary ceiling. Some argue that the virus will eventually reach a stable equilibrium. Scientists are cautious with this assumption. Stability is context-dependent. Changes in human behavior, immunity, environment, or technology shift the equilibrium continuously. What appears stable now may not remain so under new conditions. The most sobering aspect of probabilistic forecasting is that none of these outcomes require failure. They do not depend on negligence, incompetence, or denial. They occur even when science functions as intended, even when surveillance exists, even when countermeasures improve. Evolution does not punish mistakes. It rewards opportunity. This reframes the future not as something to be prevented entirely, but something to be navigated. The question shifts from how do we stop the virus from evolving, to how do we design systems resilient enough to absorb ongoing change. This is a fundamentally different challenge. It extends beyond virology into governance, economics, and ethics. Probability also demands humility. Predictions are not prophecies. They are distributions. Scientists do not claim certainty about specific outcomes. They identify ranges of likelihood. Within those ranges, preparation becomes the only rational response. Surprise is not eliminated by knowledge, but vulnerability can be reduced. What happens if it evolves again is therefore not a single answer. It is a spectrum of futures weighted by likelihood. Most are manageable, some are destabilizing, a few are severe, none are impossible. The danger lies not in imagining the worst-case scenario, but in ignoring the most probable ones because they lack drama. The virus does not need to evolve into something unrecognizable to change the trajectory of human health. It only needs to continue being what it already is, slightly better adapted each time. This is the quiet logic that unsettles scientists the most. Not fear of the unknown, but recognition of the predictable. In this sense, the future is not shaped by surprise. It is shaped by accumulation, by small advantages compounding over time, by systems strained gradually rather than broken suddenly, by a threat that remains just manageable enough to persist indefinitely. This is why scientists speak less about endings and more about trajectories, less about victory and more about sustainability. The virus will evolve again because that is what biology does. The real question is whether human systems evolve with the same consistency, foresight, and patience, because probability does not wait for readiness, it only waits for opportunity. Section 10. The choices that decide the outcome. At this stage, the trajectory is no longer determined by biology alone. The virus will continue to evolve according to rules that have existed for millions of years. What remains undecided is how humanity chooses to coexist with that reality. The outcome diverges not because the virus changes, but because societies do. The most consequential variables now are not mutations or variants, but policies, priorities, and collective tolerance for long-term risk. One of the first choices is whether to treat persistent viral circulation as a temporary anomaly or a permanent condition. This distinction shapes everything that follows. Systems built for emergencies behave differently from systems designed for endurance. Short-term responses prioritize speed and visibility. Long-term responses prioritize resilience, redundancy, and quiet effectiveness. Confusing the two leads to fatigue without resolution. Public health infrastructure reflects this choice more clearly than any speech or declaration. Surveillance programs that are continuously funded, updated, and normalized signal acceptance of permanence. Systems that are activated only during crises signal denial. The difference is not technical capacity, but political will. Monitoring a persistent threat requires patience, and patience rarely produces headlines. Another defining choice involves how risk is communicated. Societies must decide whether honesty about uncertainty strengthens trust or undermines it. Simplified narratives offer comfort but degrade credibility over time. Complex realities, when acknowledged consistently, allow populations to adapt expectations. The decision to speak plainly about trade-offs rather than promise closure determines whether trust compounds or erodes. Economic policy becomes a central arena of divergence. A virus that causes widespread but uneven impact exposes structural weaknesses. Some sectors absorb disruption easily, others accumulate damage quietly. Governments must decide whether to treat these losses as acceptable externalities or address them as preventable costs. This choice shapes inequality more than the virus itself. Workplace norms represent another fork in the road. Flexibility, sick leave, ventilation standards, and remote options are not simply conveniences. They are evolutionary pressures applied to transmission dynamics. Societies that normalize health-preserving behaviors reduce opportunity for spread without mandates. Those that frame accommodation as weakness inadvertently favor persistence. Education systems face similar decisions. Schools are not just sites of learning. They are nodes of transmission and adaptation. Investments in infrastructure, air quality, and hybrid models influence long-term outcomes. The choice is not between safety and normalcy, but between short-term disruption and chronic instability. Healthcare systems, too, must choose between reactive treatment and proactive prevention. Treating illness after it manifests is visible and politically safer. Preventing illness requires sustained funding, early intervention, and acceptance that success looks like absence rather than achievement. This is a difficult narrative to maintain in democratic systems optimized for immediacy. The allocation of research funding reveals priorities with particular clarity. Investments in variant surveillance, pan-pathogen vaccines, and broad-spectrum antivirals signal preparation for continuity. Funding limited to reactive development signals hope for finality. Neither approach is inherently wrong, but each commits society to a different future. Another pivotal choice concerns global coordination. Viruses do not respect borders, but responses often do. Data sharing, resource distribution, and synchronized policy reduce evolutionary opportunity. Fragmentation increases it. The decision to prioritize national advantage over collective stability reshapes the global risk landscape. Cooperation is not altruism in this context. It is risk management. Information ecosystems also play a decisive role. Societies must decide whether to treat misinformation as an external nuisance or an internal vulnerability. When trust fractures, even accurate data loses influence. Addressing this requires more than correction. It requires consistency, accountability, and institutional credibility built over time. Cultural attitudes toward illness influence outcomes in subtle ways. Normalizing presenteeism, stigmatizing caution, or framing vulnerability as personal failure creates conditions favorable to transmission. Cultures that decouple identity from productivity allow individuals to make health-preserving choices without social penalty. These norms evolve slowly, but their impact is cumulative. There is also a choice about how success is measured. If success is defined solely by reduced mortality, long-term morbidity is overlooked. If success is defined by hospital capacity alone, population-level wellness erodes unnoticed. Expanding metrics to include cognitive, cardiovascular, and functional health changes the conversation and the policy response. Political systems face the challenge of time horizons. Election cycles reward visible action and immediate relief. Persistent biological threats demand planning that extends beyond individual terms. Choosing to insulate public health institutions from political volatility increases continuity. Failing to do so ensures oscillation. Another decision concerns equity. Viral persistence disproportionately affects those with fewer resources, less flexibility, and higher exposure. Addressing this disparity reduces overall transmission. Ignoring it concentrates risk in predictable populations, where it eventually amplifies. Equity is not a moral add-on. It is an epidemiological strategy. Societies must also choose whether to invest in adaptability rather than prediction. Forecasting specific outcomes is inherently uncertain. building systems capable of absorbing surprise is more reliable. This includes modular healthcare capacity, scalable data systems, and rapid response research frameworks. Adaptability reduces the cost of being wrong. There is a deeper philosophical choice beneath all others, whether to frame coexistence with evolving pathogens as failure or as a condition of modern life. Framing it as failure encourages denial and exhaustion. Framing it as a condition encourages design, innovation, and acceptance without complacency. The divergence is subtle at first. Small decisions accumulate quietly. Budgets are trimmed or sustained. Programs are extended or allowed to lapse. Language shifts from precision to reassurance. None of these choices feel decisive in isolation. Together, they determine whether the future is constrained or resilient. The virus will continue to test these choices repeatedly, not through dramatic escalation, but through persistence. It will expose gaps slowly. It will reward consistency and punish neglect. This is not vindictive. It is mechanical. The outcome, therefore, is not written by the virus. It is written by how societies structure their response to uncertainty, fatigue, and inconvenience. Biology sets the parameters. Humanity chooses the path within them. There is no single correct decision. There are trade-offs, costs, unintended consequences. What matters is whether decisions are made consciously, with awareness of their long-term implications, rather than by default. In the end, the future diverges not at moments of crisis, but at moments of calm, when attention fades, urgency dissolves, and choices become easier to postpone. That is where trajectories are set. The virus will evolve again. Whether its impact deepens or diminishes depends less on what it becomes, and more on what humanity decides to be.